Good morning, one and all. Welcome to this webinar series. Before we start the next session, let us just have instructions that is needed to carry on with this webinar. Kindly do not chat as soon as you log in, giving greetings and introducing yourself. Your name will be correlated with your mail ID in registration form and will be recognized accordingly. Do not ask questions as soon as the sessions begin. You can ask questions half an hour after the start of every session around 11 a.m. You will be prompted to ask questions at the end also. So you can type your questions then. Your participation in all the 3 webinar sessions is important for us to be eligible for the certification. So kindly log in before 10:45 a.m. as the chat room will be locked after the set time. End of the session, the feedback form link will be posted in the live chat on YouTube. So it is mandatory to fill the form for every session to be eligible for the e-certificate. So we seek your kind cooperation for the success of this webinar. Thank you. Have a great session.
A very good morning to one and all. Welcome to the second session of this webinar series organized by the PG and Research Department of Biotechnology, Women's Christian College, Chennai, on global research trends on COVID-19, opportunities and challenges. I would like to welcome today's guest speaker, Dr. Ashok Kumar from the Genetic Engineering Department of Madurai Kamaraj University. A warm welcome to you, sir. Thank you for accepting our request to be the speaker of the day. Let us all bow our heads to the Almighty God. Dr. Julia Harriet Sumati would lead us in prayer, followed by Dr. Anita Ajay Singh, who would introduce the speaker to you. Dr. Anchana Devi of the department will handle the question answer session. Thank you. Namaskar.
Loving Lord Jesus, we thank you for this beautiful day. Thank you, Lord, for your grace and favor, which was on us on the first day of the webinar. Lord, we commit the second day into your hands. Lord, we know that you will lead us and guide us. We thank you for all the participants who have registered. May they have an urge to learn new things. Lord, I pray for the speaker for today. Thank you, Lord, for connecting him with us. Thank you for the knowledge bestowed on him. Thank you for the years of expertise in his field. May he deliver a fruitful session, which will be very useful for the student community. I commit the entire session into your nail-pierced hands. May we have a smooth session today. We give you all.
It gives me immense pleasure to introduce our resource person for today, Dr. B. Ashok Kumar. He is an associate professor in the Department of Genetic Engineering, Madurai Kamaraj University. He holds a doctoral degree and in his field of specialization are molecular genetics, genomics, and genetic engineering. Dr. Ashok Kumar has a keen interest in areas such as congenital disorders associated with nutrient metabolisms, microbial bioremediation of xenobiotics, probiotics to alleviate diabetes and hypertension. C. elegans as an animal model for vitamin transport physiology. Dr. Ashok Kumar has a rich professional experience spanning over 23 years. Through these years, he has had a number of national and international collaborations with institutes such as University College London, UK, University of California, USA, Indira Gandhi Institute of Child Health, Bangalore, Institute of Nanoscience and Nanotechnology, Gandhigram Rural Institute, Gandhigram, NIMHANS, Bangalore, CCMB, Hyderabad, and IIT Kanpur. He has completed 8 research projects and has 3 ongoing projects in hand. He has several honors, awards, and recognitions to his credit, which include the ICMR International Fellow Award for Young Biomedical Scientists for the year 2016-2017. Dr. Ashok Kumar has over 60 publications and presented several papers in India and abroad. He has organized a number of conferences, seminars, and workshops. He has co-authored and published over 7 books. He holds memberships in a number of professional and academic bodies in India and abroad. Dr. Ashok Kumar has 2 patents to his credit. A renowned research guide, Dr. Ashok Kumar has held several challenging administrative posts at Madurai Kamaraj University. With all his work and responsibilities, Dr. Ashok Kumar has consented to join us today to talk to us about the diagnostic tests for COVID-19 daunting challenges. Over to Dr. Ashok Kumar.
Good morning. Good morning to one and all. Thank you so much, ma'am, for the nice introduction. At the outset, I'd like to thank the organizers for providing me this opportunity to share my research experience in molecular genetics. During this lockdown due to pandemic corona infections, the technology advancements through Zoom, is facilitating a lot of student youngsters to educate themselves. It's one of the greatest opportunity for the students to utilize their time judiciously, reasonably for educating them. Thanks for the opportunity. Actually, just a minute. Yeah. I'll be sharing the title of my presentation. Am I audible to you? Excuse me?
Yes, sir.
Okay.
You are audible.
Yeah.
Audible, sir.
Thank you. All right. The title of today's presentation is Real-Time PCR in COVID-19 Diagnosis, Prospects and Challenges. Right. So before starting my talk, I'd like to give a brief introduction about this COVID-19 and its unique features about it. There are many viral infections caused serious mortality to humans, but in what way this COVID-19 is different from other similar kind of viruses. This coronavirus, which exhibit high frequency of mutation during replication, especially deletion mutations are quite prevalent during replication. They do also undergo high frequency of recombination during replication. This is quite unusual for an RNA virus because this coronavirus, which is very smaller in size, which is about 65 to 125 nanometer in diameter, which is having positive standard RNA as its genome. The size of the genome is somewhat 26 to 32 kb in length. Single standard RNA containing virus. Okay. A kind of a high frequency recombination is very unusual, a kind of RNA virus, especially for non-segmented virus. What do you mean by non-segmented virus? The genetic material, if it lies in a single genome without any partitions, it is said to be non-segmented. If it is scattered in a sense, basically they are said to be segmented. But here, all the necessary material which lies there in the nucleic acid RNA alone, a single genome containing virus. And because of this high rate of recombination, there could be possible for a lot of variations in the genome of the virus, which makes irresponsible when we target to control these viral infection with various drugs now. They may not be responsive because of these variations. Structure-wise, genome-wise, which is quite similar to the SARS infection, which happened earlier due to CO1 around 2003 and 4. But there are quite good percentage of differences in their genome also. Similar to SARS-CoV-1, in this virus also, human airway trypsin-like protease, cathepsins, and transmembrane protease, shortly they are called as TMPR SS2, are essential for splitting virus once it is attached with its receptor, and which help them to spread to other cells, which is also exactly present in the SARS-CoV-1. If you talk about the structural characteristics of this virus, the main compounds that we need to consider are spike proteins, end protein, membrane protein, and envelope protein. Spike protein of this virus contains two subunits, S1 and S2, which is responsible for the binding with the receptor ACE2, and S2 domain, which is responsible for the fusion of this virus with the membrane. N protein plays important role in the pathogenesis, replication, and packaging of the matured viral RNA into the capsid. Membrane protein is the most abundant structural protein, which is ornamenting the viral envelope. Is a glycosylated protein. Envelope protein is nothing but another small structural protein which is expressed in the infected cells. It plays key role in the morphogenesis of the virion within the cell. Right. So, statistic I would like to give here. Until yesterday, what happened? So far, how many countries are affected with this COVID infections? Totally, they are 216, and 54 lakhs of cases are confirmed so far, and almost more than 3.5 lakhs deaths have happened. Such a debilitating, disease-causing virus. We need to know how to control it. Many deaths have happened already, but these infections caused death rate, which is classified based on the age. Which age group of people are mostly vulnerable to this viral infections? 80 plus above. Those age peoples are more vulnerable to the viral infections, which are accounting almost 15% of death all over the world. But it's very surprising to notice that the age group between 0 to 9, the rate of fatality is very, very rare. At least we should be happy with that. In addition to that, all the comorbidities also noticed in the people who have died because of these viral infections. So in the list, cardiovascular diseases occupies the first place. Cardiovascular diseases, diabetes, chronic respiratory diseases, those who have hypertension, in cancer. So these people are more vulnerable to the viral infections and they are dead. So in that part also, we should feel happy that normal persons, they may have these kind of viral infections, but, so due to the immune response of such hosts, the viral infection may evade slowly. But those who are all having cardiovascular diseases, diabetes, and other disorders, so they are facing serious issues with these viral infections. And next things. So we know that through aerosols and to human-human interactions only, these virus is getting transmitted. Through nasal entry, it is getting infecting a human being. Once after infection, what's really happening there? And how many days you need to see the impact? So once after infection, there is a window period. That period may be asymptomatic from day 0 to 7. That period we call it as window period. In that period, you may be asymptomatic, or one or few symptoms you may see, or you will have onset of symptom in that period. From 7 to 14 days, you will be able to see the release of immunoglobulin, the major immunoglobulin IgM, which is becoming detectable in the infected patients as a response against these viral infections. Okay, they will start to increase in your blood and will start to decline in the day 21st. From the image, you will be able to see that. Later, from 14th to 35th day, you will be able to see the other antibody IgG. IgG. This Ig, which tells you that when there is a release of IgG, in a sense, you can say it's a convalescent period. Okay, so this is what the immune response you will normally see in an infected person. What is the need of understanding this image? So once after having a viral infection, within a week period, you may not be able to see any kind of response. If you go for a diagnosis in that period, the detection of antibodies as a response of a host against this virus may not be detectable, because there will not be any antibody against the virus. But after 7th day, you will be able to see the presence of IgM antibody. Then what you will be able to see in the first 7-day period? You will be able to see live viral particles or the viral genomes. If you have a kind of such a period in a sense, in the period, instead of looking for antibodies, if you focus your attention to see or to detect the viral particles or the viral nucleic acid RNA, then you will be able to diagnose, because that stage is quite active stage of viral infection. Right. So, for example, in a period, if you see IgM, the next slide will easily help you to understand. If you are able to see IgM in a sense, the second column, patient may be in the early stage of infection, because the immediate response from a host will be the release of IgM against any antigen. In the later convalescent period only, you will be able to see IgG production. Okay, right? So again, I will go back. So this is how you will be able to decide what kind of diagnosis method you are going to follow to diagnose the COVID-19 infections. Right. So here in this slide, I have given you the strategy of using variety of diagnostic techniques. Earlier, we were depending on mostly the antibody-based blood test because this is quite easy and simple too. Very less time consuming. But unfortunately, in India, when we do this kind of antibody-based diagnosis, there were many false positive. That's a separate story. We don't need to worry about it. Immediately, if you suspect a person who is having a kind of symptoms like cough, cold, fever, and sore throat in a sense, the first thing we have to do, quarantine the person, and during that period, we will be collecting nasal swab. You can also take blood sample from the infected patients, and you will be able to do antibody testing. If you are positive for that antibody testing in a sense, your clinical assessment tells you that you are infected, then you may go for a treatment with the drugs after isolating yourself. But still, if it is negative, but he is showing symptoms, in that situation, we should go for RT-PCR, real-time PCR analysis to detect the nucleic acid of COVID-19. Why? Because you may be in the early stage of infections. At the stage, you may not be able to see antibodies. So the viral particle just got inside to your cells, and these viral capsids are broken out, and within the nucleic acid which was present within the nucleic acid has come out now, and it will undergo early transcription, other issues, and maturation, late transcription, and finally, rupturing the cell, and you will be able to see the release of urea. But once infection has started, in a sense, your RNA will be released in your cell and will start to go for a replication. So in that stage, you will be able to see the presence of viral nucleic acid alone. So in that period, if you do a test using antibody testing, you may not be able to see any positive results. So in that period, the right method you have to choose real-time PCR analysis to detect the viral nucleic acid. If you are positive with that RT-PCR, then you have an infection, okay? If you are negative, but still you are showing the symptoms of cough, cold, and other symptoms, you are in a condition to be tested continuously. After few days, again, we will be going for real-time PCR or antibody-based testing. If PCR is not at all done, but still, after 15 days, there is again a possibility to go for antibody testing or real-time PCR. Thus, you will be able to really confirm whether the viral infection have happened because of this COVID-19 or not. So this is the normal flow chart is being used for the diagnosis of coronavirus infections. In the next slide, and I have shown you already, once you do real-time PCR, qPCR, if you are positive in a sense, patient may be in the window period of infection, early stage of infection. You are positive to real-time PCR and IgM in a sense, patient may be in the early stage of infection. If you are positive for real-time PCR, IgM and IgG testing, patients is in the active phase of infection. Positive for real-time PCR and also for IgG, patient may be in the late or recurrent stage of infections. If you are positive for only IgM, patient may be in the early stage of infection. RT-PCR results definitely would be the false negative. There could be more chances for having false negative. If you are positive for only IgG, in a sense, patient may have had a past infection and he has completely recovered. So this is the clinical significance of the following testing. If you are positive for IgM and IgG, patient may be in the recovery stage of infection. Maybe the RT-PCR results would have been false negative. Right. Now we'll jump into what is the real-time PCR and what is the technique, how it helps for the diagnosis of viral infections. Real-time PCR is a very sensitive and a reliable method for the detection and quantification of nucleic acid, either DNA or RNA. Their levels also can be quantified. Hence, these real-time PCR techniques are generally known as qRT-PCR. Often, the real-time PCR is confused with the RT-PCR. RT-PCR is nothing but reverse transcriptase PCR. But here, real-time PCR it's purely meant for the quantification of nucleic acids. Either it may be in the form of DNA or RNA. If it is DNA, straight away, you can use that DNA as a template for your quantification purposes. If you are having RNA that need to be converted into cDNA by RT-PCR, then that cDNA need to be taken into consideration for further quantification. Here, during the amplification, the products are produced at each and every cycle. But how it is being detected? That amplification, DNA amplifications are detected in terms of fluorescence emitted by a reporter molecule which is added in the buffer which is used for real-time PCR. Variety of fluorescence materials are used for these kind of purposes. So those signals will be emitted when you go for each and every PCR cycle. During PCR, we will be setting a PCR for minimum 30 to 40 cycles. So each and every cycle, your template DNA will be amplified, your double-stranded DNA will be amplified. 1 will become 2, 2 become 4, just like that. In your folds, you will be able to see amplification. Here, in the real-time PCR analysis, you will be able to see, once after PCR amplification, you will be getting a PCR product now. At every single cycle, you will be getting product. The formation of the product will be monitored in a real time. Hence, we call this methodology as a real-time PCR. In a normal PCR, once after the completion of your PCR, you will check out the PCR finished sample and you will be loading into a gel and you will be using ethidium bromide to stain your DNA. Then you will visualize your DNA in the gel after doing agarose electrophoresis. But here in this methodology, you will be able to monitor while the process is going on. When the DNA amplification process is going on, in that time itself, you will be able to see when the amplification is started, what was the period of lag period, there was any amplification that, no amplification in that period. Then you will be able to see exponential phase or logarithmic phase. your DNA will be started to amplify. For that, it will require some cycles, maybe 12th cycle or after 14th cycle or 16th cycle. Somehow you will be able to see an amplification that will be there in an exponential phase. After some cycles, it will reach a plateau. So there will be a saturation. You will be able to see saturation in its amplification. But the log phase is the most crucial phase where actively the DNA doubling is going on. Okay, that can be monitored during the amplification in a real time. Hence, this methodology is said to be real-time PCR. Okay. So where this technology is being applied, for what all the applications, real-time PCR is useful. For example, if you have your RNA as a template, your qPCR can be used for gene expression profiling analysis. For example, you have a normal condition, you are growing one particular cell or a bacteria. At the same time, similar bacteria or same bacteria, which is incubated at 50 degrees Celsius. Due to this variation in temperature, there could be a lot of changes in the expression of different genes. You really want to quantify that kind of expression. Expression in a sense, your RNA need to be, your DNA need to be first transcribed into RNA, and then this transcribed RNA will undergo translation process. Thus, you will be able to see one particular gene is expressed or not. So for any kind of expression analysis, your starting material should be an RNA, So you have to take RNA from that cell from two different conditions. If you suspect the particular gene should be differently expressed in a sense, you have to use specific primers to amplify that particular gene. And you will be looking the expression pattern of that particular gene in two different conditions. Yes, bacteria which was incubated in normal temperature. At the same time, a bacteria which was incubated at 50 degrees Celsius. What happened to this particular protein? For example, heat shock protein, HSP. What happened to this particular heat shock protein when you incubate at 50 degrees, whether it is abundantly expressed when it was incubated at 50 degrees Celsius or not, that can be quantified. For example, a normal person and another person is having a cancer. So cancer is there will be an involvement of multiple genes. So what kind of genes are overexpressed in a human who is having a cancer? If you take RNA from the cells of cancer, of the cancerous person and a normal person, you will be able to study the whole expression of variety of genes. RNA expression profiling can be done. In a simple PCR, you will be able to do single gene analysis. Each and every gene will be analyzed and then you will be able to go for it. But at globally, gene expression profiling can be done. A sophisticated technologies like microarray and high performance of RNA profiling machines are available now. With that, you will be able to understand the whole expression profiling of the genes in normal condition and at the diseased condition. For these kind of purposes, your real-time PCR is helpful to confirm whether it is really differentially expressed or not. Likewise, microRNA profiling also can be done with the help of real-time PCR. MicroRNAs are small non-coding RNAs are the key regulators that control gene expressions. They are non-coding RNAs. You will be able to understand the pattern of those microRNAs by real-time PCR. For that, you need RNA as a template. Okay. With the DNA, what else can be done? Single nucleotide polymorphism genotyping. Whether is there any polymorphism in that particular gene or not, that can be studied. An allelic discrimination can be performed, whether this particular SNP is in homozygous state or heterozygous state, because we have two alleles, two copies of genes we do have. whether we have mutation in only one allele or it is there in both the alleles. If it is in only one allele, you will say heterozygous. If it is there in both the alleles, you will say homozygous. For that kind of a discrimination, your real-time PCR helps. Mutation, somatic mutation analysis also can be done. Copy number, how many number of particular gene is present in the cell, that also can be quantified using real-time PCR. Chromatin immunoprecipitation quantification can be done. DNA methylation. DNA methylation is a very important process of controlling gene expression. If there is any promoter, if it is methylated, the promoter of the particular gene cannot be expressing. Okay, whether that particular promoter region is methylated or not. If you really want to test, you can use a real-time PCR. Pathogen detection, whether the particular bacteria, whether it was there or not, whether the particular virus, whether it is there or not, if it is having DNA as its genome. For these kind of applications, real-time PCR helps you a lot. All right, next. A kind of an introduction about the history of real-time PCR. Higuchi in 1992, the first one who did real-time PCR analysis and also demonstrated the process of real-time PCR analysis. That time he used yttrium bromide. We know that it's very carcinogenic. To replace that, nowadays we are using so many fluorescent dyes that we will discuss in later. Right. So this kind of a PCR is also called as kinetic PCR because you are continuously measuring the increase in the intensity of your particular DNA. Because there will be a camera that will be helping you to capture the emission of fluorescence, that you will be able to understand, okay, there is a PCR amplification is going on. All right. So for doing this real-time PCR analysis, in 1996, ABI Biosystems, Applied Biosystems is the company. First, they devised a machine, okay, ABI Prism. Okay, nowadays there are a lot of versions are available. Those images I've given you here. Real-time PCR analysis from BioRed also I have given. So many advancements we have with these machines. Few of the examples I have given you here with. So these machines are equipped with performing PCR reactions. Once after the reaction is done, the emission of fluorescence material will be detected in your cameras, and those images will be captured, and thus it will be analyzing, okay, whether the PCR amplification is going on or what it is the stage the amplification is happening. Everything can be measured in a machine. Right. Okay. So now I would like to say something about the methodology and how many types of real-time PCR is there. There are two different types of RT-PCR, real-time PCR, sorry. There are two different types. They are, one is said to be absolute quantification by real-time PCR, and the other one is relative quantification by the real-time. What do you mean by absolute quantification? Here, you will be able to determine the quantity of your gene of interest by its own amplification, by comparing with the standard curve of each and every single target. You are not just going to compare with anything. You will be counting or quantifying the exact quantity of the particular nucleic acid is present. Hence, it is said to be absolute quantification. With the nucleic acid, whatever you have, when you go for real-time PCR, it will be giving you three phases. The amplification will be giving you three phases. That image I will show you later. A lag phase. There is no amplification here in this phase. Later, exponential phase. It's a very active dynamic phase of amplification. And finally, saturation. Right. So when you have an exponential phase, in a sense, in that stage only, real amplification is happening. But you do not know how much of your initial input as nucleic acid you have given. To know that, you should have a known quantity of your template with the known copy number in different dilutions. Using that different dilutions, you should plot a first standard graph. Once after making the standard graph, with your unknown sample, you will be able to exactly find out what is the quantity of nucleic acid of your own interest or your own viral DNA or RNA it is present or there. You will be able to come to know by this methodology. CT is nothing but a threshold cycle. At what threshold you are able to see preferential amplification of your DNA, your logarithmic phase of amplification of your DNA. For that determination only, your CT value will help you. What do you mean by relative quantification? Exactly similar, but here for its comparison, you will be using any one of the housekeeping gene. For example, 18S rRNA, GAPDH, beta-actin, because these are all the genes are housekeeping genes. Any kind of physiological stress when you give, their expression will not be different. But certain gene expression will different depending upon the environmental conditions. But here, these gene expression will not be differed. So they are called as housekeeping genes. If you really want to quantify a particular gene of interest, in a sense, you will be comparing the CT value of your unknown sample with the CT value of your known housekeeping gene. Thus, you will be relating what is the expression level of your gene of interest in relating with the housekeeping gene. Thus, it is said to be relative quantification. So, totally, there are two different methods of real-time PCR. Absolute quantification, you will be exactly quantifying, but here you will be comparing with the housekeeping gene. Thus, you will come to know that what is the load of or what is the copy number of your gene of interest is there. Right. The next one. As I said, absolute quantification and relative quantification. For the relative quantification purposes, generally, cyber green is the fluorophore is being used. Cyber green is a cyanide-based dye, which will be emitting light at 520 nanometer, green light. So this will be able to detected by your detectors. So this is the dye which is commonly used for relative quantification purposes. Similarly, whatever you did with already ethylene bromide or acridine orange, it's just an intercalating dye. This intercalating dye will bind only with a double standard DNA. You can see cyber green, yeah. Will give a green fluorescence at 520 nanometer. So this dye emits prominent fluorescent signal when it binds with a minor group of DNA non-specifically. It's a non-specific. So you will be doing this analysis in comparison with one of the housekeeping gene, either 18S or beta-actin or GAPDH, anything can be used. Okay. The second one is Tagman probe. Tagman probe is very selective and specific method which is used for absolute quantification of gene expressions. Tagman probe. Here I have given you the image of a Tagman probe. What is it? It's nothing but a DNA probe, a small stretch of DNA probe of 14 to 16 nucleotides in length, which is specific to the minor group of a DNA region. In this probe, the 5' end of your probe will be added with a marker, a fluorescent dye or reporter. Hence, it is shortly called as R. The 5' N will bear a reporter dye. Commonly, what we are using, fluorescent, which is shortly called as FAM. In the 5' end of your small probe. In the 3' end of the same probe, you will have a quencher. Quencher, commonly what we are using, tetramethyl rhodamine, which is added in the 3' end of your DNA. What's really happening over here? Normally, when you have high energy quencher, your reporter will not emit light when it is in close proximity. When it is in close proximity, your reporter dye will not emit light because of the presence of the quencher when it is in close proximity. If they are hydrolyzed, if they are removed, if they are broken into pieces, then the released reporter will emit light. This is the principle of Tagman probe. Tagman probe is a hydrolysis probe which is specific to a particular gene position of your DNA, particular DNA position of your gene. Especially, always it is highly designed to go with the minor group of your DNA. In such a way only this will be designed. One of the end added with reporter, another end added with quencher. When it is in intact contact, even though if light is emitted, even though if it is light is applied on its probe, this will not emit light because you have a quencher that has quenched the emission of the light. You will not be able to see, or you will not be able to detect. During PCR reaction, this probe need to be hydrolyzed. This probe need to be displaced. When it is broken, that time your reporter dye will be released out. If it is released in a sense, when you pass the light on it, now this will be emitting light because quencher is not attached. How it is happening? That's the technology. I will show you now. Now you can look at Tagman probe detection. If you really want to detect a particular gene of a virus or bacteria, whatever it is, for its amplification, normally you will be designing primer, forward primer and reverse primer. They will be gene specific or DNA specific primers, right? While you perform this PCR using Tagman probe, along with the primer, you have to design a Tagman probe. That Tagman probe, as I said, 14 to 16 base per nucleotide with the fluorescent dye, fluorescent and the quencher. It is now added over there. This will be binding with the single standard DNA of your interest. Whatever the gene you are trying to amplify, when you design primer, you have to design a probe also. When you look at the image, you can see the forward primer and reverse primer. Now, in the PCR tube, you should have one particular enzyme. We call it as ampli-tag DNA polymerase. Ampli-tag DNA polymerase has three properties. One is 5' to 3' DNA polymerization activity. Second one is 5' to 3' exonuclease activity. Third activity, 3' to 5' proofreading activity. But here, the second one I said no, 5' to 3' exonuclease activity. What will be happening? When you do PCR, you will be annealing the primer binding. At annealing temperature, there will be primer binding. Primer has bound over there. At the same time, your Tagman probe also bound with your DNA template. Whatever the DNA polymerase added, it will be start adding new nucleotides depending upon whatever which is there in your template. The complementary nucleotides will be added one by one depending upon whatever you have the template. When the cycle continues, it will be reaching the probe whatever you have already added. Now your 5' to 3' exonuclease activity will chew the Tagman probe. So this will be breaking the Tagman probe into pieces. When it is broken now, the polymerase reaction will continue. The released broken Tagman probe, now fluorescence is already attached over there now. That will be broken, which is separated out of your quencher. Now this will be emitting light. So your detector now will detect emission of the light. In a sense, first PCR cycle I have started and gave you an amplification. That is what you will be able to keep in your mind. So once you see the detection, in a sense, the first probe has been eliminated, hydrolyzed from the reaction, and now it is emitting light because earlier it was quenched. Then the cycle continues. Then you will see more number of fluorescence emission, in a sense, more number of cycles are happening. Thus, you will be able to see whether the amplification is happening in real time or not with the help of Tagman probe. Tagman probe, which is designed for the minor groove regions, and they are attached with one particular chemical which is called as dichlorocyclopyrrolidine tripeptide, which is used to fuse or conjugate this with the Tagman probe, because this will be facilitating very tight binding. Right. So this is how the Tagman PCR, real-time PCR works. But differently, cyber green, you will be able to see. This will be able to bind only with the double-stranded DNA. When there is a melting, your double-stranded DNA will separate and primer will be binding and new double-stranded DNA will be synthesized. When new double-stranded DNA is synthesized, wherein the intercalating cyber green dye will go and bind, will give more fluorescence. This is the principle for cyber green detection, and this is the principle for Tagman probe detection. Now, we are setting up Tagman probe PCR reaction. For that, what else you need? You need a template. That may be a viral DNA. In case of COVID-19, we are now currently detecting either S gene, N gene, or E gene. Whether presence of these genes, N gene, S gene, and E gene, are being detected by the technology currently available. Thus, we are able to say, yes, you are infected with corona or not. So the initial thing is you need template. How much of template you need for this kind of reactions? Minimum 10 nanogram. Minimum 10 nanogram, maximum 1 microgram, 1000 nano. Maximum 1 microgram of DNA or RNA you need for these reactions. All right. And primer you need, forward and reverse primer, the probe, Tagman probe you need. And you have a master mix. That master mix contains tag DNA polymerase, magnesium ions, DNTPs, buffer, and passive reference dye. What is the need of magnesium ion? Magnesium ion is a cofactor for tag DNA polymerase. This will be increasing the fidelity of your binding of your DNA and increased amplification you will see. If you do not have magnesium ions in your buffer, you will see very, very faint amplification of your DNA. But if you add magnesium, you will be able to better amplification. So normally 2 nanomolar of magnesium ions are being used. If you use more than that, there will be more chances for non-specific amplifications. Always it is recommended to use 2 to 5 nanomole of magnesium chloride ions in the buffer, which is nothing but a cofactor for your tag DNA polymerase. Right. Thus, you will be performing this Tagman Pro PCR in a single tube, we call it as one-step PCR, or in 2 stages, we call it as two-step PCR. What is the difference between this? Because you are using here RNA. This RNA need to be converted into cDNA by reverse transcriptase. That cDNA only is going to be used as a template for further cycle of PCR with your tag DNA polymerase. So this can be done together in a single tube when it is said to be one step. You can do separately. Conversion of RNA to cDNA is one step, and using that cDNA as a template for doing real-time PCR analysis and another step. This is two-step or two-stage PCR. Either way, you will be able to do. Currently, whatever we are practicing in Tamil Nadu is a single-step PCR for the detection of COVID-19. Especially, we are doing this COVID real-time PCR for the detection of N and E gene presence. All right. Once after performing experiments, you will be able to see this kind of graph. Now you can see in one side relative fluorescence you have. There I have given you CT, threshold cycle. At the range only, you will see the progressive amplification of your DNA. The detection, whatever the fluorescence detected now, you will be able to see the logarithmic phase of thing. There, blue one is nothing but low quantity of template they have used. The green one is the greater quantity of template they have used. The red one is unknown quantity. Just by comparing with the blue and green, you will be able to understand what is the level of red, the unknown one. Thus, you will be able to understand the viral load, how many number of viral particles are there in a patient that can be detected. For that, you should do known copy number DNA with the real-time PCR. When you perform, you will get some numbers. With the numbers, you have to perform or you have to plot a standard curve. And from the standard curve, you will be able to come to know what is the quantity of your viral DNA you have in the patient, that you will come to know. So this is how your final output of your real-time PCR will be there. All right. So with that, I thought of closing the very basic information about real-time PCR analysis, mechanism, methodology, types, how it is being performed, and how do we diagnose COVID-19 infections, this and that. But there are more chances. This is 100% accurate, but still there are many limitations with this technology. You have more chances to have false positives. You need to be very careful with the false positive. You see a person who is having all gold standard symptoms in him, but still if he is negative by real-time PCR analysis, in a sense, he need to be quarantined and he need to be watched for some more days, to test again, either by antibody testing or real-time PCR analysis. Then only we will be able to come to a clear conclusion about it. There are many chances to get false positive due to manual error. Sometimes the machines may lead to some other issues while making reactions. If artifacts by bubbles, if it is introduced while doing PCR reactions, you may not be able to see proper amplifications. Those limitations also need to be considered in our mind while we performing these experiments. But this is a very perfect, 100% accurate, fast and reliable diagnostic methodology currently being extensively used for the diagnosis of COVID-19. There are so many viral drugs are in practice. Earlier, the anti-malarial drug hydroxychloroquine was suggested to be consumed by everybody who have got COVID-19 infections. And yesterday, US President Donald Trump, he said that I stopped consuming this hydroxychloroquine because of side effects. And people are also saying that it is non-effective. Why we are using hydroxychloroquine for treating viral infection, because this is meant for malaria. Why we are now started to use it for COVID-19? Because, when COVID-19 infection starts, okay, so when it is attaches with the lung epithelial cells, it is getting invaded by invagination. Your virus is getting, first it's absorbing with the ACE2 receptor, and it is getting invaginated. Thus, it will be forming an envelope like an endosome within the cytoplasm of the infected cell. That endolysosomal bodies need to be lysed out, then the viral particle will be released. How this is happening? Because of the changes in the pH of the entire endosome. Normally, the pH of the endosome will be neutral. When the pH changes to acidic, your endosome will be lysed and your viral particle will be released. But what your hydrochloroquine does, this will be increasing the pH of the endosome. If the pH becomes alkaline, your endosomes cannot be lysed. So thus, your hydroxychloroquine will be interfering with the breakage of your endosomes, and thus it will be halting the replication of virus. That is what it is prescribed now to treat COVID-19. But we are unsuccessful, and at the same time, due to the concern of side effects, the consumption of hydroxychloroquine has been now stopped by WHO, but still it is being prescribed in our country. All right. In combinations also, it is also being recommended. And other drugs, remdesivir and favipiravir, variety of cancer drugs also are being repurposed for the coronavirus infections. With that, I thought of closing this presentation, and thanks for your patient watching of this presentation. If there are any questions, please feel free to ask me. Thank you.
Thank you, sir, for
Yes,
your interesting and informative presentation. And I am sure that students would have got a clear view on the diagnostic approaches of COVID-19. So as you talk, it was a complete package, I should say. Like, it started from the basic principle about the structure of the coronavirus and explaining why the virus is not responsive for viral drugs, explaining about the window period, its clinical significance, and testing it. And as such, the explaining the basis of principle of RT-PCR and up to its application, and closing with the information about the drugs currently used. So now we'll move on to the question answer session, sir.
Sure.
So kindly can you stop sharing your screen?
Yeah.
Thank you.
Is it stopped now, ma'am?
Yes, sir.
Okay.
So participants, kindly post your questions in the live chat along with your name and institution name. So I have got few questions on roll.
Sure.
The first question is by Aprajita Chakraborty, PhD biotechnology student from Xavier's College, Calcutta. She wants to know if the RT-PCR test is false negative. Is any alternative test available to test whether the person is positive for COVID?
Yeah, right. So as I said, immunotesting can be done using antibody. Antibody testing can be done, but we need to wait for some time. If you are negative, false negative by real-time PCR analysis, so the person need to be quarantined for a few more days. And later, after 1 week, again, we can perform the same real-time PCR analysis or antibody for the detection of antibody IgM tests can be performed. Thus, you will be able to come to know whether he has an infection of COVID-19 or not. So superior techniques are available. Whole genome sequence is available, but it is very costlier. The affordable is point of care devices using antibody testing. Very simple technique. We will be able to use that to detect.
Okay. The next question, sir, is from Rebecca from Global College. She's asked, is PCR better in diagnostic finding of COVID-19 than the test like X-ray and CT scan? About its reliability and
Very confirmative test. Real-time PCR analysis is the confirmative test, which is the best methodology worldwide, which is being currently used. That's the reliable technology. People are trying to develop other technologies like antibody detection, antigen detection, but still there are many failures people have noticed while compared to this. So it seems to be RT-PCR technology seems to be the highly reliable methodology for testing COVID-19.
Okay. The next question is by Bijita from Lady Bourbon College, Kolkata. She has asked, what is serology testing and how it is effective for COVID-19 detection? About different type of serology testing methods in COVID-19. So how it is effective? I think she refers to the kit test.
Right. So that depends on the specificity of antibody, whatever we are using. See, as I said earlier, viral specific IgM detection at the early stages. If we have a right antibody to detect those produced IgMs. Sometimes in the infected patients, you will be able to see the presence of viral antigens like spike proteins, envelope proteins. So these proteins are known as antigens. If you have an antibody against that one, you will be able to use that also to detect. So they are also reliable, but poor quality of or otherwise, so if it is monoclonal antibody, whatever the antibody they are using for detection, if it is monoclonal antibody, in a sense, you will be able to expect very better results. Nowadays, right now, we have not spent much time to use monoclonal antibodies. Right now, we may be using the polyclonal antibodies. Polyclonal antibodies will be maybe giving you other cross reactions, so that may be interfering with our observations, but still, they are also good methodologies. I came to know that a couple of days ago, Indian government is trying to check the quick methodology of testing using antibodies. Because right now, if we are taking a nasal swab sample, in a sense, those people who are working are getting contradicted with the viral infections. So that number is increasing like anything. Instead, if you withdraw blood, in a sense, so with the syringes, then you will be able to go for testing with this kind of immunologically testing methods. So which is being highly advised nowadays, instead of going with real-time PCR analysis.
So next question is by Venkateshan Govindaraju from Hindustan Arts and Science College, Chennai. At which phase the COVID-19 virus is detected?
Once after infection. If you are sure you are yesterday infected, today you will be able to see. But the viral number of virions will be very less in 1 or 2 days of infections. At least within a week period, you will be able to see good number of virion particles. Then it is quite good to have a nasal swab, and with that, whatever the virions you have now, with that you will be able to get good quantity of your RNA template. Then that can be used within a week, which is so good. But that can be detected from first day onwards.
Okay. The next question, sir, from Samira Khatun from Jabas College for Women. At which phase or stage these antiviral drugs are prescribed? At which phase or stage these antiviral drugs are prescribed?
In even some cases, for the prevention purposes also, these drugs are being used. Because some of the viral drugs which do interfere with their replication process. Now, once after infection, for example, as I said, hydroxychloroquine. So this will be preventing the infections during the formation of endosomal invagination. That period itself, this will be interfering in essence. In the very early stage of infections only, this can be administered. Even in the late stages, if it is administered, further progression of the disease only can be prevented. Further serious infections because of the viral spread, that can be prevented. But even at the early stages, these drugs can be taken if you are suspicious to have a viral infection.
Related to the same question, sir, Shubhada from National Institute of Virology, Pune, she asked how HIV drugs work against corona. There are some scientists claiming that due to hydrochloroquine causes more death.
Yeah. Yes. So because of that reason only, the HIV drugs are also being administered because they are all antiviral drugs. So they could be interfering with the RNA-dependent RNA polymerase, which is a viral-coded enzyme that is essential for the replication of this virus. So those drugs, they do interfere, they do inhibit that enzyme. So because of that, replication can be prevented. That's the ultimate target of these drugs. So that purposes only, why don't we repurpose the drug, whatever we have created for HIV. So we are trying to attempt and we are trying to show whether are you able to see any protection when we use HIV drugs or not. So yes, it is being recommended. HIV drugs also being recommended.
Next question, sir, from Rahul, Mr. Rahul, Christu Jayanti College, Bangalore. He wants to know what are the chances of getting false positive results by the RT-PCR method. False positive results by RT-PCR method. What are the chances?
Okay, right. So the early stages of a sample collection, very early stages of sample collection, the viral load may be very, very less. Very, very less. Improper transportation of the materials. So once after the viral nasal swabs, that need to be left in a viral transport material. That transport material should be in good quality. Otherwise, so it may lead to a lot of disintegration of the viral particles, So that need to be transported properly to the lab. And from there, there are methodologies to isolate. Guanidine isothiazide, trizol, which is being used for RNA isolation. If you are not good in isolating RNA from the sample, in a sense, okay, if your RNA, because it is highly labile material, if it is denatured, you may lose your RNA samples. So you may have a low purity RNA sample. When you use that low purity RNA sample for your real-time PCR analysis, you may not be able to see proper amplification. And man-made errors, pipetting, at the same time, sometimes the air bubbles will be introduced as an artifact. That will be interfering with the amplification. That may not be properly recorded by your cameras when it captured the fluorescence emissions. These are all some of the reasons are responsible for the false negative results of real-time PCR, as far as my knowledge.
Okay. So next question is by Ashok Kumar from MITS School of Biotechnology. Will COVID-19 stop ever? Because this virus keeps mutating it, and many scientists say that this mutation is the main problem for making vaccines.
Yes, absolutely. So far, 220 genomes of viral, this COVID-19 virus has been genome sequenced, including 2 viruses from India. A lot of variations, they have seen it. Lot of variations. From that, they will be able to come to know that at least there could be 10 to 13 different types of variants. So these variants are clustered together into 3 forms, A, B, C. Just like that, in the Middle Asia, a particular form of virus is quite common. In the European region, or one particular virus type of which is so common. In the China, a kind of viral viruses are quite common. So that kind of classification has been made into. This is the one of the reason why we are not able to see vaccine immediately. For example, we've been trying to generate vaccine for dengue for a long time. Still we are unsuccessful. There are many vaccines are available for many viral infections or bacterial infections. We were successful. But with the dengue, it is not at all positive. The reason is same. Likewise, COVID is also similar kind of virus. We face problem, but shortly we will find a solution for that.
Okay. So next question is by Pratika from WCC. How do we relate the rapid antibody test with asymptomatic carriers? Does cross-reactivity play a role here? So that's the question, sir. How do we relate the rapid antibody test with asymptomatic carriers? Does cross-reactivity play a role here?
Yeah. Similar kind of, similar kind of viruses also may lead to these kind of symptoms. Symptoms are asymptomatic, So we need to keep monitoring them, keep watching them. Then we will be able to further performing either serologically or real-time PCR based analysis we have to do. So there is no other way to go.
Okay, sir. The next question is from Kalesh Selvi from Bharatidasan University. Our question is there any other instrument which can be used to quantify the COVID-19 virus, that is coronavirus? Any other quantification apart from RT-PCR?
See, people are working right now without using PCR at isothermal conditions. That means a single temperature conditions. There is a technology, isothermal amplification of DNA. So for that, you don't need a PCR machine. In the PCR machine, you have to set annealing temperature, you have to set extension temperature, you have to set denature. We don't need to do like that. You have to have your template DNA. You mix all the reagents. You keep at a room temperature. Then at a particular single temperature conditions like 42 degrees Celsius or 45 degrees Celsius, that is more than sufficient to amplifying something. For that, you need to have a specific requirements. There is a kind of technology, you do not require a PCR machine, then you will be able to still see the amplification. So it is there.
Next question by Amrita Sahu, Osmania University from Hyderabad. What are the chances of a fetus getting affected if the mother is positive for the virus? Is there any way to find out if fetus is infected?
So far, no reports like that, but it is possible if we have amniotic fluid sample, then similar kind of diagnosis can be performed. Yes, it is possible, but so far no reports.
The next question is by Surabhi, Central University, Jharkhand. So his question is carrying out RT LAMP process for COVID-19. Is this process similar to QT-PCR, or this process is having quick results than other PCR?
Somewhat similar, but I cannot say completely it is same. Yes, the same principle is also applied over there, but the reagents and other components, whatever we are using for this LAMP PCR is different from a routine QRT-PCR.
So participants, we are flowing with questions. So we'll take another 3 or 4 questions on roll to sirs, and we'll wind up the session. So kindly refrain from it. So next question is by Lokesh from Laila College, wants to know vitamin C and zinc sulfate are given for the treatment against corona. So will they give good results on recovery rate?
Yeah. Preliminary results are showing progress. So they will be helping for lysing the viral particle, the zinc. Yes.
Next is by Premi from Jabas College. What is the maximum recovery time for patient infected from coronavirus?
It varies from 10 to 14 days. Recently, I came to know that people from Singapore, they have got cured within 10 days.
Okay.
See, that could be reason because of the variant, which one infected the people. One virus could be less variant, the other one may be more virulent, because the variant could be reasonable for the virulence of these viruses. So maybe that could be the reason for that. But people have seen it.
So next question is by Dr. Rachana Bhavan, Vivekananda College. Any treatments that can potentially bring down the death rate? I mean the preventive loss of life.
Not getting what?
Any treatment that can potentially bring down the death rate, that means prevent the loss of life. Any potential treatment is available right now?
Cannot be exactly sure. In combinations, people are trying to cure. Many people are almost, how to say, more than 20 lakh cases have been already permanently cured. But I was looking yesterday, I was looking for that record, the number, but I was not able to get it from WHO's website. But I was searching for that. So how many numbers so far got cured because of treatment? But I was not able to. But it is there. A combinatorial therapy could definitely give a permanent cure.
The last question, sir.
But in future, we have to wait and watch. Then only we will be able to answer for this question.
Yes, sir. The last question. SK Kolam from Vidyasagar University, West Bengal. His question is, are children also at risk of infection and what is their potential role in transmission?
Yeah, everyone is vulnerable, but the fatality, whether the rate of fatality is very, 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 very less in the age group 0 to 9. That's very good news for us. I don't know what is the reason behind this. There could be many explanations for that, but still, so children are vulnerable for the viral infections, but their fatality rate, mortality rate is absolutely less.
Yes, sir. And one last question, what are the kits now that we are using for the RT-PCR? Are we procuring it from some other countries, or we are using our own products, or some information regarding the kits which has been used in RT-PCR, sir?
Many of the research institutes in our country and the universities, RT-PCR facility is one of the very basic facility. So we do normally purchase from abroad, variety of companies, ABI, Roche, Biorad. These people are selling RT-PCR machines. But almost all the institutions and universities will be definitely have. It's one of the earlier PCR was the basic facility. Nowadays RT-PCR has become the very basic facility.
So right now we are procuring it from foreign countries.
Sir? Yeah, absolutely. We don't have our own country-made real-time PCR.
Okay, thank you for patiently addressing all our questions put forth by our participants and explaining them very clearly, sir. And now we have come to the end of session 2 of the webinar series. So on behalf of the Biotechnology Department, Women's Christian College, Chennai, I wish to extend my heartfelt thanks to our resource person, Dr. Ashok Kumar, for his presentation, which was loaded with interesting information. We are also grateful to you, sir, for taking your time off to share your experiences with us and patiently answering the questions of our participants. We thank our participants for staying throughout the program and bombarding us with questions. And indeed, it's a very encouraging gesture to us. We extend our sincere thanks to our principal, Dr. Lillian Jasper, and WCC management for their constant support. We wish to thank our student volunteers who stood by us throughout the conduct of the webinar. Thank you all. And we request you to kindly join us again on Friday, 29th May 2020 for the next webinar by Dr. Ramasamy Rajesh Kumar, Senior Scientific Researcher, College of Agriculture and Biotechnology, Zhejiang University, China. He is also a founder and director of Syn Biogenica Private Limited in India. So he will be addressing the students on the topic, birth of biotechnology industries for life science students, aftermath of COVID-19. He will be focusing on various career opportunities for the life science students post-COVID scenario. So stay tuned at our YouTube channel for the next upcoming session. Thank you all once again. Stay safe, stay home, wear mask, and keep social distancing. Thank you. Have a good day.
Thank you. Thank you.